Dear student, now we will discuss about why urinary estriol provides a good index of fetal well-being. The fetus, mother and placenta, they are in close cooperation for steroid hormone biosynthesis in the fetus and placenta. They behave almost as a single functional unit. The fetus, the placenta and the mother, it behave as a single functional unit, and this unit is called the fetoplacental unit. And this unit, it helps in the steroid hormone biosynthesis in the placenta and the fetus. So how the steroid hormone is synthesized, that we know, just a bit recap. Steroid hormones, they are synthesized from the precursor cholesterol. It is converted to pregnenolone, and this pregnenolone will be converted to progesterone with the help of 3-beta hydroxysteroid dehydrogenase. Now pregnenolone can be converted to 17-alpha hydroxypregnenolone with the help of 17-alpha hydroxylase, and these hydroxypregnenolone can again be converted to 17-hydroxyprogesterone with the help of 3-beta hydroxysteroid dehydrogenase. So progesterone or 17-alpha hydroxyprogesterone, their formation, it depends upon this enzyme 3-beta hydroxysteroid dehydrogenase. Now 17-alpha hydroxypregnenolone can be converted to dihydroepiandrosterone with the help of this 17-20 lyase. And dihydroepiandrosterone, ultimately they will form the sex steroid, whereas progesterone will form the mineralocorticoid, and 17-alpha hydroxyprogesterone will form the glucocorticoid. Now we want to see what happens in this fetoplacental unit. So this enzyme 3-beta hydroxysteroid dehydrogenase is a very important enzyme because it converts pregnenolone to progesterone or 17-alpha hydroxypregnenolone to 17-alpha hydroxyprogesterone, and both of them are required for the synthesis of cortisol and corticosterone. The interesting point is the fetal adrenal gland. So this is a fetoplacental unit. This part is a fetal unit. The central part is the placenta, the intervening unit between the mother and the fetus, and this portion is the maternal unit. So fetal adrenal gland, it lacks this 3-beta hydroxysteroid dehydrogenase enzyme. As a result, the fetus depends for the progesterone. For the supply of progesterone, it totally depends on the placenta, and this progesterone will help them to synthesize the aldosterone, the cortisol. So fetus depends on a placenta for the supply of this progesterone. Fetus, however, it can convert the pregnenolone to dihydroepiandrosterone. So it is a kind of sex steroids. So it cannot convert pregnenolone to progesterone, but it can convert pregnenolone to dihydroepiandrosterone, and in the fetal liver, this dihydroepiandrosterone will be converted into 16-alpha hydroxylase dihydroepiandrosterone. So both are the precursor of sex steroids. Now the placenta, on the other hand, it lacks this specific enzyme, that is 17-hydroxylase or 17-20 desmolase, which converts the pregnenolone to dihydroepiandrosterone. So it therefore depends on the fetus and also from the mother for the supply of this dihydroepiandrosterone. So placenta, it gives progesterone to a fetus. Now fetus and the mother, they gives dihydroepiandrosterone in return to the placenta. Now in the placenta, this dihydroepiandrosterone, it is converted into the estrogen. So dihydroepiandrosterone can be contributed by the mother, but this 16 alpha hydroxy dihydroepiandrosterone, it is exclusively converted by the fetus. Now when this dihydroepiandrosterone, it is converted into estrogen, then it will form the estriol in the placenta, and this estriol will come out via the maternal urine, and 90% of urinary estriol in mother during pregnancy, it is from the estriol derived entirely from this fetoplacental unit. So if we measure this estriol in mother's urine, we can get a good index of the fetoplacental unit functioning or the fetal well-being. If the fetus is good, if the fetoplacental unit is working properly, then the estriol amount in the maternal urine will be normal. So it explains urinary estriol provides a good index of fetal well-being. Thank you and subscribe. For reference, you may consult Ganom figure number 22-23 at 24th edition.